Hi, welcome to the BioLab. So you come in, you'll see that we have assortment of PPE, personal protective equipment. One of the main important things about being in the BioLab is be safe, essentially. So we have some gloves here. You can also find gloves in the store. We have lab coats, and our lab coats are going to be behind this door here. We also have safety glasses. Safety glasses are important if you're pouring any corrosive substances like, bleach or something into a cup. You want to make sure you have safety glasses on so it's not splashed to your eyes. We also want to make sure we have appropriate attire. So closed-toed shoes are important. It'd be nice to have longer pants, longer sleeves. If you have long hair, make sure you pull it back so that it's not falling over into your experiment. You also want to make sure we have no food or drinks in the biolab. We also want to make sure we have a clean space. Make sure you leave it clean, just as you see it, or even better. So that means putting away chemicals, putting away materials, putting away equipment. So MakeHaven is a BSL 1 facility, meaning that it's a biosafety level 1. Biosafety level 1 is suitable for work involving well-characterized agents not known to consistently cause disease in immunocompetent adult humans and present minimal potential hazard to laboratory personnel and the environment. So if your material is within BSL1, you're good to go. But if it's not within BSL1, please consult with the facilitator and we can talk about your project. I'll take you and show you some of the equipment we have and things that you can use them for. So on the section here, we have our autoclave and our heat vacuum desiccator. We have our pipettes here. We have glassware. We have a sink. There's also a drying rack for our glassware. Please make sure you empty this so it will get filled up with glassware if you just wash your dishes and put them here, but please make sure you put them back onto the shelf if you use glassware. We have our large centrifuge that's also refrigerated. We have the acid and base cabinet here underneath the centrifuge. We also have some common chemicals and solutions that you could use. Some of them are buffers, some of them are electrode solutions for calibrating the pH electrode. We also have salt and sugar. This is not for eating, this is only for lab purposes. And we have a couple more buffers that might be useful for you. Here we have our biosafety cabinet that you can use to keep a sterile environment or protect yourself or your sample from, the ambient environment. We also have in this container here just extras of petri dishes, pipette tips, pipettes. So if you need something, try looking through there, see if we have it. Here we have incubators. So these two incubators here, one is kept at 35 degrees Celsius, another is kept at 29. You can change the temperature, but please let a facilitator know why you're doing it and how long you need to do it for. Over here we have an assortment of equipment. So we have two centrifuges. They're smaller ones. We also have a tube rotator. We have a magnetic stirrer. We have a PCR machine, and we have a smart dock, which is used for visualizing gels. We also have a scale here, a microwave, and a microscope. We also have a nanodrop, minus 20 degrees Celsius freezer here. We have a stir plate. We have a vortex genie, which you just use to mix up your solutions in a tube. We have a sonicator, and there are two hot plates that you can use to put little tubes in and keep your samples at a particular temperature. We have tube holders. There's electrophoresis set up here. There's a pH electrode. And then we have our fridge here. So one of the most important things about being a lab in a shared lab space especially, and also just being a good scientist, is to label everything. So if you're creating a solution and you're putting in a buffer, make sure you actually label that what that solution is. So, say that it's H2O, say your name, your full name, Stacy Uchendu. Then you can also put the date that you made that solution and then the expiration date. So sometimes it might be 30 days, sometimes it might be 28 days, it might be never. But make sure you put down the name, date, and what that solution or substance that you're using is so that we can ask questions about it if we need to move it around or dispose of it later. Speaking of disposal, if you're using biohazardous things, such as, like, let's say you're doing an experiment with a petri dish and you had some bacteria in it, some approved bacteria, BSL-1 bacteria, and you wanted to get rid of that petri dish. So there are a few ways to do that. So for solid waste, such as a petri dish, you want to actually autoclave it first. So you would put your petri dish into a autoclavable tray or an autoclave bag, put it into an autoclave, and that deactivates any of the germs or bacteria or anything that you're working with, essentially. And then once you have it autoclave and it's safe and it's cooled down, you can put it right into the regular trash. And if you had, let's say, a solution of, some type of BSO1 safe bacteria, and you had it in a beaker with, like, water or something else, or a buffer, then you can dispose of liquid waste by using 10% bleach solution. So we have some bleach down here. Make sure you wear some safety goggles, 10% bleach, put it into the beaker, let it sit there for about 10 minutes or so that it can really deactivate the hazardous components in it. And then you can pour it down the drain with a copious amount of water. So just pour it down the drain while the water is running and let it run for a minute. And then when it's done, wash up your glassware, put it away. If you want to get rid of liquid waste, you can also autoclave it. You can only do one or the other. Do not put bleach in something and then put it into an autoclave because you could create a toxic chlorine gas. So if you just choose to autoclave without bleach, that's fine. If you choose to bleach without autoclaving, that's fine, but one or the other, not both. We also have a sharps container down here. So if you're using needles or pipette tips or maybe even like some glassware that broke, you can put them down there into the sharps container. We also have some chemical storage places. The first one is the shelf here. So these are where we have chemicals that really don't pose any risk to your safety or health. So we have salt, sugar, some buffers. These don't have to be in any particular cabinet, so you can just put them here. If you have an acid or a base, you can put them into our acid and base cabinet here. And if you have something that's flammable, you can put it into our flammable cabinet here. The important thing to note for these acids, bases, and flammable materials or reagents that you're using is that you want to have a MSDS, which is a material safety data sheet. And you want to put that into the binder here. So in case someone, you or someone else has it on their skin or in their eye or whatever else, you want to know what kind of how to take care of that situation and what kind of risk that might pose to their health. So if you're getting any acids or bases or flammable materials for your own personal use, make sure you put your name, your date, and the substance of what it is, and make sure that the MSDS sheet is in the binder. To make sure that you have the date that you put that into the cabinet, because it can be there no longer than 1 year. If it's there past 1 year, it's going to get discarded. You should be able to discard of your materials by yourself. That is your responsibility. If you have any questions about how to discard an acid, base, or flammable material that you store in the cabinet and you're not sure how to do it, please consult with the facilitator and we can put a waste disposal plan. But the ultimate responsibility is with you. If you have any questions, you should consult the Biosafety Lab workbook that's online. You'll see some more guidelines there, especially about what kind of things you can bring into the lab. Otherwise, happy science.